Matthew, I was hoping that you'd be able to introduce yourself and provide an overview of your research regarding short chain fatty acid production.
Yeah, so what we do in the lab is we use an in vitro batch fermentation approach where we could take human fecal samples derived from different compositions, and we could screen nutrients using this type of in vitro batch approach. From there, we do a bunch of different analyses, whether we sequence the DNA to get a good idea of where these compositions are shifting, but at the same time, you could spin down the plate, remove the supernatant and filter it, and then we could run it on a GC to get a good idea of the metabolic output that these communities put out in response to glycoproteins and carbohydrates. And we could actually use this method to screen other fibers as well, pectins, soluble fibers, prebiotics such as inulin, and use that as a good method to screen compounds.
That makes sense. Would you be able to talk about why GC-MS is the right type of analysis for you?
Yeah, so traditionally with volatile molecules such as short chain fatty acids, since they're volatile, GC-MS seems to be the standard to go for analyzing short chain fatty acids. The literature also uses, I think there's new methods. You could use HPLC, liquid chromatography as well. They're trying to move toward a type of assay where they have some sort of conjugated, not antibody per se, but something that could bind to short chain fatty acids, but the sensitivity and the accuracy of this type of assays aren't great. So traditionally, it's been GC-MS or in some cases HPLC.
Very good. And I know you touched on it briefly, but could you get into the sample preparation process that you're using?
Yes, unlike human fecal samples, which we also do, we have to ship those off. They're very dirty. They have a bunch of stuff in them. So instead, by using an in vitro batch approach, we use a 1% ratio of our fecal sample. We add it into this complex medium, add our nutrient that we want to add. In our case, we add glycoproteins. And then after the fermentation process, these small communities produce these metabolites. And then we'll spin down the plate for about 20 to 30 minutes at about 4,000 RPM. And then we'll filter it out using a 0.22 micron filter. And then we're ready to rock, and we pretty much run the samples from there.
Very good. What are the implications in terms of your research for better understanding human health and dietary interventions?
That's where the field gets really fun and complicated, since right now there's thousands of metabolites are being produced by the human gut. We know that short chain fatty acids are the major molecules that are being produced, and they have a lot of implications for human health. By using targeted metabolomics, we're able to get a good idea of how much of these short chain fatty acids are being produced. I think in the future, it'll be a lot nicer to see what metabolites we might be missing by using an untargeted metabolomics approach. And maybe we could start to narrow down specific metabolites that contribute to certain metabolic diseases. But right now, using the targeted approach, we could get a better idea of what fiber structures seem to work better for what compositions. So since human gut microbiomes are different, there's some microbiomes that degrade certain fibers better than others. And using GC-MS or chromatography as an approach, we could get a good qualitative as well as quantitative idea of how much metabolic output is happening by these gut microbial communities in response to a given fiber.
I know you touched on this, but do you see trying to integrate more HPLC in your work?
I do. I think that would be in terms of looking at untargeted metabolomics. There was a beautiful paper, a protocol paper that just came out of Stanford at Dr. Sonnenberg's lab that showed that they're using an HPLC approach to do untargeted metabolomics, and then they compare that to a reference database of untargeted metabolites. And it opens up the view per se of how many metabolites we could actually look at with high accuracy and high identification percentage versus having to set up a methodology for targeted metabolomics, and you only get so many. So I think the HPLC route might be the future of metabolomics and how they interact with epithelium and human health and disease.
It'll be interesting to hear how this progresses and I would also think, and correct me if I'm wrong, but your sample prep needs might change based on that switch pretty considerably.
I figure they would, and especially what type of sample we use, whether we want to collect human fecal samples or even mice samples, the cleanup would have to go through the roof compared to in vitro batch approach where we're using a low sample biomass. I would agree with that.
A little bit of background on my equipment at Organomation. I would connect about 90% of our blowdown evaporators to LCMS, GC-MS, or HPLC. So that very much is our business in terms of the types of research and analysis that Organomation equipment is supporting. Thank you, Matthew. I appreciate it.
Absolutely.